me, and then I can let you know directly. But for the greater audience, that might be a great question that we can answer in the River Report. So with that, we're going to move into the science Q&A. I think I'll just go back through some of the questions that came in specifically for the people that already spoke. So Lawrence, if you're okay, I will jump back for that person who sent you a question back when you were speaking. I'll also stop sharing my screen. Well, Lawrence, I wrote some comments when you were talking, and I was like, wow, the integration, I had never imagined that you could have a button on your fishing rod that would then result in the data of the moment being captured. That's really amazing. And of course, the scientists want access. As soon as you're talking about this data, I'm like, how do we get access to this data so we can understand these systems better? But the question that was asked was, are these apps transboundary, and can they be used to manage stocks across borders? So similar to the eBird app that looks at bird migration.
That's a good question. Now, I know, you know, the Angler app. I'm getting a little echo here, but okay. That did it. The Angler app, My Catch, done by Anglers Atlas. It really, you have to have some regional sort of appeal. If you make it just too generic, then people are going to say, well, how does this relate to me? And how does this relate to my region, the waters I love? So a lot of them are doing is you can create custom skins for them. You have the basic app, which has the functionality, and they can constantly upgrade that functionality. And then the look and feel of the app itself can be tailored to meet a certain regional flavor, the species of fish, the times, the information about seasons and waterways and so forth, local resources, links to local resources, all that can be customized to give the app a special regional flavor. And then at the back end, who gets access to that data, how it's shared, what databases does it land in, that can all be tailored as well, right? That's another whole set of connections that can be tailored to fit the interested parties. So for sure, it can go to resource databases around the world. There's no reason why it can't. It's just a matter of who wants to have access to that. If you're doing redfish studies in Florida, do you really want to know about muskies on the St. Lawrence River and vice versa? So there's those types of things to be need to take into consideration, as well as the variables that you're tracking. And at that point you're almost into customizing the engine of the app itself. But building these things, that can cost easily $100,000, and even customizing an app to give it a regional flavor can get up to $20,000 quickly.
Wow. Well, thank you, Lawrence. That's important to hear. We were also talking about building linking apps to the River Report. And yes, I've spoken to you and Kat Kavanagh in the past, and developing apps is a costly business, so it has some challenges. Lawrence, I'm going to move on to Jeff. There's a couple of questions for you, Jeff. So in your representation this morning, you showed sediments along the Cornwall waterfront had the highest mercury concentrations. What is happening with these sediments? Is there a program in place to deal with them? And do high water flows that we've had in the most recent years move the sediments around?
Wow, nicely. There's lots of questions there. Who else do we have in this panel? Maybe I can swing it to some of those people. Some of these, I can get a stab at it. So
we also have Alison Derry, who's joined us, and Matt Windle. It's myself and Marianne, and otherwise it's on you, Jeff.
Okay, well, that's great. So I'll choose my flavor there in terms. So the first question was, first part of that question was something about the highest concentration of sediments along the Cornwall waterfront. Is there a program in place?
That's right. Is there a program to deal with them?
Yeah, actually, it's a great question, because certainly, if you remember those slides, this is the source of mercury. The major point source of mercury was located at Cornwall. I mean, there's mercury sources throughout the Great Lakes and from atmospheric deposition and whatever. But really, I mean, as that slide, if you recall, showed that there was a real sort of concentration along the Cornwall waterfront and then downstream, showing that this was really an important source at Cornwall. And we do know, of course, that we're industries that were using mercury and tons of mercury, in fact, that was used and lost to the river. So, but again, as the highest concentrations along the Cornwall waterfront, it really did result in a great deal of assessment, and there was it was one of the major items that was on the to-do list when that issue became, you know, through the sampling became aware that this was the problem, and so through a review process, the question was, you know, is this mercury, are those sediments active? And is it affecting the benthic invertebrates that live in the sediments along the Cornwall waterfront? And based on those two questions, the answers were yes, the mercury is, and took some research. It was both government and academic research that showed that the mercury, the sediments containing that high level of mercury, was getting buried gradually over time. And so what we found is, as if you started at the very top level, the mercury levels were lower, and it was it was basically getting lower as you moved up towards the surface. And so that meant that there was a layer of cleaner sediment that was separating and isolating the sediments that were deeper down. And the other thing was we found that the benthic invertebrate communities were not grossly affected. In fact, they were not significantly different than reference sites. And so that those two major issues suggested that that really the sediments were stable, and that the approach was put in place. It's called the Cornwall Sediment Strategy, which was the most important thing is not to disturb those sediments. So there's a process in which if someone wants to do a project along the Cornwall waterfront, they have to contact the Conservation Authority, Reason Conservation Authority, that implements a process of review and understanding of those projects, location, that sort of thing. So, so that's the program in place. It's called the Cornwall Sediment Strategy, and it's an important part of maintaining that status where the concentrations are maintaining the ordering in terms of the cleaner sediments on top of the lower ones. So that's the program in place, and in these sediments are, the other part of the question was whether they're getting moved around with the faster currents.
Yeah, with the faster currents.
Yeah, the issue there is that these sediments are located in embayments, in areas that are shielded from those currents. So as the currents change up and change over with these recent conditions, those areas are not as affected. But that's still a good question, and there's actually a study underway led by the government agency to look at the stability and ensure that these higher currents aren't really actually making any difference. But that's the impression we have right now.
Okay, thank you. That's great. I'm also going to take a moment because it's been confirmed that we have our full panel with us. So along with Matt Wendell from the River Institute and Alison Derry from UQAM, we also have Jerome Marty from the Canadian Academy. So I welcome the three of you here. Of course, the three of you didn't talk this morning, so if anyone in the public would like to address questions to them, please do that as well. Otherwise, I'll be throwing questions out and hoping that anyone on the panel will pick them up. We had a question opportunity open on the website for a while, and Kathy McRae sent in a question that says, my family has had a cottage in Somerstown since 1940. Over these years, we have seen many of the reed beds die and disappear. We wonder why this is happening. Kathy couldn't join us today, but she asked if I would ask the question and then relay the answer to her. So if someone cannot take that one, I would be grateful. Who would like to take it? Anyone?
Well, it's Jeff here. I'll get started, but I think I'll throw it also to Jerome and to Matthew. I think should chime in, and maybe Alison as well. I mean, the issue around weed beds, the big issue is the fact that we've seen reductions in the amount of nutrients that are available since the 1970s and 60s. And so through the Great Lakes Water Quality Agreement that I spoke about, there's been a real emphasis on decreasing the amount of phosphorus, decreasing, remember those phosphorus in detergents back in the day. And so as those nutrient levels, that is the fertilizer essentially for those weed beds, decline, the tendency for those weed beds to grow up the way they used to decline. But there's other factors there too. I don't know whether or not Jerome or Matthew or Allison would like to maybe address any of those questions.
Yeah, I can add a few words. Can you hear me?
Yes, we can hear you well, Jerome. Go for it.
Very good. So yeah, to add to Jeff's comments about the declines in nutrients, phosphorus and nitrogen, I think depending on where we are on the river, there is also other questions around erosion and the dynamic around that. That in some areas, probably we are losing different forms of habitat, including weed beds, but in other areas it's improving. So it's always when we think about especially a river and it has flows that's been managed in some areas and less managed in others. But all of this makes it a very dynamic ecosystem, and it does change over time. Since 1940, so that's quite a while back. And
yes,
it's true that since 1940s, there's been a lot of nutrients that has come to the river because of all the activities in the watershed. The watershed was likely a lot less developed in 1940s than what it is today. So yeah, it would be interesting to see if there was a curve here where with increasing land use, we saw a potential increase in plants, macrophytes, but not only macrophytes, also algae. And now, because of the decline of such nutrients, we should see a decline in the biomass of plants as well. So some good stories, I guess. What I'm trying to say is that we do have good stories to report about the decline in nutrients, but it does have consequences for other things. So we need to keep an eye on all these elements.
Yeah, and I really like this question because it spoke to me after I mentioned in my talk about Ian McIntosh taking us out to show us his images that had been taken 100 years ago with all this emergent vegetation, and then taking us back and showing us how that had changed. So I thought of that when I read the question. Matt, was there anything you wanted to add?
Just Jerome did a great job touching on the erosion factor and the water level issue, and where Somerstown is between two dams that control the water levels to a very small margin of variation between Cornwall and Montreal. So from the 1940s, you would have had very large fluctuations in water just naturally occurring from spring to fall. And now what's happened since the late 1950s is that this water level has remained fairly stable. And then you add on top of this the issue of variations in amounts of nutrients declining significantly, and also just ship traffic coming through Lake St. Francis as well, and some of the erosion issues that Jerome would have talked on as well. So you're maintaining a water level that's very, very stable now for 60 years essentially, and all these other factors on top of that. So vegetation beds are definitely going to adapt to that and definitely change dramatically compared to, say, the 1940s for sure.
Yeah, great. Well, I'm sure Kathy.
Lee, it's Lawrence. I know a lot of people point their finger at common carp as being a major disruptor of weed beds when they forage around, in nearshore areas. But common carp have been in the river for over 100 years. So I think pointing the finger at one species of fish that a lot of people love to hate, it may not be it may not be a good idea. I mean, we're scapegoating them, but at the same time, if we are having fewer and fewer weed beds because of nutrients reduction, and yet the common carp population has been stable for over 100 years, does that mean you got more fish focusing on a smaller part of the river to forage on, and maybe that's accelerating reduction? So I mean, it's something that needs to be looked at for sure.
It's exactly these comment, this input that you give, Lawrence, all these different perspectives that I think helps to round out the, the discussion. And it's some of this that we're trying to bring together with the River Report as well, right? All these observations that people are making and then trying to link them to how does it make sense from a science perspective. So yeah, thanks for adding about the common carp. I have some more questions, and I want to go after asking one about one that was rooted 80 years ago. I want to move to one for the future. We've gone through a big process for the River Report of identifying indicators, and we have our set of indicators. And even as we have this set and we're trying to get the data together for this set, people are asking us about the next set that we're going to develop, because in our set we don't have what the next emerging threats are. So some of the threats that are coming, we've basically made an assessment on the indicators that we're doing on the threats that we know about. So Alison Derry, I wanted to ask you this question. Can you comment on what you think the next emerging threats to the river are?
Okay, well, some of the emerging threats that we'll be addressing in university programs supported by NSERC are microplastics. And so this is really an emerging area of concern that's being identified as a priority area by our scientific funding body for natural sciences and engineering in Canada, NSERC. And so a group of researchers are presently putting together, kind of like Montreal University is putting together a grant application to deal with microplastics in the St. Lawrence River, and in partnership with the River Institute as well, and specifically addressing, for example, how microplastics contribute to bioconcentration, bioaccumulation, biomagnification of contaminants in the food web that are, for example, absorbed on those plastic particles. So as they pass through the digestive tracts of animals, not only may they physically interfere with feeding and behavior, but there's substances stuck on the plastic, such as toxic metals and organic toxicants such as PCBs and flame retardants that may enter the tissues of animals and biomagnify up through the trophic levels up to fish that humans consume. And so this is very important, especially since virtually nothing is known about it in freshwater systems. The base research has been done in marine systems, but even there, we don't really know how this type of pollution is influencing the food webs. So that's definitely one example of an emerging threat, including, I suppose, others like flame retardants that are going in with the influent plus absorbed on the particles.
Thank you, Alison. It's so good to hear your answer, because I was in my previous life I was out west at the University of British Columbia, and I was involved in teaching. Well, I was the teaching assistant for a marine pollution course, and this issue about the plastics, and not just actually eating the plastics, but the contaminants that adhere to them, had been raised back then, and it's it's probably over 10 years ago now, and I hadn't seen much of it in the freshwater literature. So it's really interesting to hear that you've got a project that's going to be addressing that. I'll open that up for comment from the panel. Does anyone else want to comment on the emerging threats? Jerome?
I can add a few thoughts. When you ask a question like this to a scientist, it's almost you're asking a personal question, because we all have our own views and based on the knowledge that we have. If I was to answer the questions, I would likely say that overall I can see that the algae, phytoplanktons, are going to take over the world again very soon, as they did early on, when the early steps of evolution. And I do think that there is a great potential with climate change for some groups of algae in particular to take over systems where they're not in yet at this point. And as we all know, not all species of algae are desirable. Some of them are having physical impact in terms of water use, but also toxic impacts when with the release of microcysteines in the case of cyanobacteria. So I think we already see these signs. We've seen them for now a couple of decades, but seeing, for example, the Rideau Canal this summer, who was literally changed into a soccer field where you can almost walk on. This was never seen something like that. It was a green fluorescent canal that we had. I can see that these situations are going to be more and more common, and we should not forget about these emerging things that we already know about them, and scientists are pretty good at tracking that. But sometimes new threats are coming up as the new fashion, and I won't criticize the plastic on that because plastic is also important. But I would hope that we don't forget about what we already know about that is certainly going on and is going to increase significantly in the future.
Thanks so much, Jerome. Yes, it is true that we all have areas that we delve into the literature, and then it comes to mind when an open-ended question like that comes up. I've got another question, and I'm off the top of my head, I want to address this to both Matt Wendell and to Alison Derry, but of course it's open for anyone on the panel. And that is the importance of wetlands in relation to biodiversity. So I think I'm going to start with, well, which one of you would like to go first, Alison or Matt, for that, the importance of wetlands for biodiversity? Matt, are you going to go?
Sure. It sounds like a question for Marianne too, Lee.
Yes.
Oh, yeah. Yes, that's true.
Marianne could do a great job answering this question as well. Obviously, wetlands are extremely important for biodiversity in the wetlands. They're per square unit, some of the highest biodiversity habitats that we have in aquatic systems, ranging all the way inside in the water up to the terrestrial system. There's just a huge wide range of different types of habitat that allow this biodiversity to take place compared to, say, more plain types of habitat you might find in large parts of the river. So very critical. These types of wetlands or habitats are being impacted dramatically, actually, through a lot of sections of the river, through development, through erosion, through water quality issues as well. And they're just really critical to protect because they act as these natural filtration systems for the river. They impede erosion along shorelines, and then they just have this really great benefit of offering habitat to so many species. And through work at the River Institute, we've documented this biodiversity. We've done surveys of wetlands all throughout the system and compared that to areas where wetlands are not located, and the biodiversity is on average much higher in the wetlands. So it's documented, and we're still conducting various surveys of wetlands. We're also using new technologies as well, like drones that capture overhead shots of wetlands, and they're evolving now that we have these new water level regimes in place on the river as well. So we've identified this as a really important thing at the River Institute to monitor over the years. So we're creating a great baseline data set of that, and really happy to work with Alison's lab on that as well for some of the fish and invertebrates science questions related to the importance of wetlands.
Thanks, Matt. Thanks a lot. I don't see Marianne's camera on, so I'm going to turn to Alison if you wouldn't mind chiming in on that question.
Yeah, sure. Yeah, so we're very happy also to collaborate with the Institute, and just to kind of add to the information that Matt also supplied, we recently published a paper that showed that wetlands not only are a source of aquatic biodiversity, but they're also performing a previously unrecognized role in locking out certain invasive species. And so in a sense, kind of contributing towards a buffering against the impacts of exotic invasion on aquatic biodiversity. And I'm referring specifically to Ponto-Caspian invaders, such as the round goby, which do not like browner water, muddier substrates. And so it is so important to hang on to these local wetlands that are scattered throughout the system, because they are at the same time maintaining high local diversity at the same time as locking out some of these very, very voracious exotic invaders that are just stripping communities at other sites. And I should add that the St. Lawrence River, I guess more in Lac Saint Louis, where Montreal is, here we have the confluence of the Ottawa and the St. Lawrence rivers. So combined with wetlands there, you also have a big ion gradient of high conductivity calcium green water flowing in from the Great Lakes, the St. Lawrence River, and the brown brown water rolling off the Canadian Shield from the Ottawa River, rich with dissolved organic carbon, tea-like substance, merging. And in this low ion water, this is also more of a macro barrier to these exotic species. So combined with these kind of macro refuges and then these micro scale refuges of wetlands, there is this buffering resistance for aquatic biodiversity against a heavy assault of exotic invasion in the system that we have not observed, for example, in the lower Great Lakes. So this is not to say that we should not be concerned about exotic invasion impacts in the St. Lawrence River. They're heavy and they're hard, and they have not yet stabilized at most of the sites. But it's a little bit of a good news story, is that a very special part of the system, there does seem to be some buffering in it to help us maintain and preserve what's left of our aquatic native biodiversity.
Wow, thank you. That adds another dimension, doesn't it? I mean, Matt was talking about just the intrinsic value and the biodiversity of a wetland, but that it also has this buffering capacity is amazing. I'm going to open it up. Marianne, would you like to comment?
Yeah, for sure. So I think I'm a little biased as a scientist who specializes on wetland environments, but I don't think that if we just look in terms of biodiversity, I don't think that these, they're not just important. These wetland ecosystems are critical for the conservation of biodiversity. If you want to see any kind of species, I suggest you go sit in a wetland and you're going to see species that you'll really see anywhere else. And if you look at where wetlands are important in organisms' life cycles, you see that a lot of animals are drawn to wetlands for reproduction and for foraging. And these two life history, history requirements are critical for, populations of these species. So I think with the historical trends of destruction and degradation of wetland environments, they not only, as I said, they're critical for the conservation of biodiversity moving forward. And I think we need to do whatever we can to protect these environments, especially along rivers where they have all these ecological services that benefit us as humans. But I really think that these, protecting these wetlands is, with climate change and all the other threats that face biodiversity today, this is one thing that we could do to help protect a lot of species on Earth.
Thank you, Marianne. That's great. That's really good. I'm going to move on, and I'm going to, Jeff, I'm going to put you back in the hot seat. You did mention the fish contaminant study with Akwesasne, and we're interested in finding out a little bit more about that. So what sort of species are being studied, and what are the contaminants that are being studied?
Yeah, I mean, we're really excited, and Matt Windle can probably chime in on this one as well. And this is a study that's actually, it's not our study. We're working in partnership with Abe Francis and the Environment Division at Mall Council of Akwesasne. And it was a really interesting, just to go back a little bit on the stories, is when we're looking at fish consumption advisories, and we started to recognize that one of the areas that wasn't sampled as frequently as other areas in the St. Lawrence River were the waters around Akwesasne, and Abe was involved in that conversation. He was like, "Whoa, this is not great, and we want that information." And he obviously is in the hot seat himself, to be able to have that data for his community. So we started sitting down, and through a series of meetings, I think we were meeting every Thursday afternoon, and you were probably part of that, Lee, for about 6 weeks, sitting down with Abe to talk about what we needed, what we would like to study, and the list got very long, but eventually, you know, and there is a long wish list, but we've eventually narrowed it down to a small number of species that have obviously are consumed frequently, such as walleye and yellow perch, bass, and I'll ask Matthew in terms of the other species that we're collecting, because he's leading, Matthew is helping to lead the field work, working with a team from River Institute and Akwesasne, and so he can provide us that update. But also, the key species, key contaminants we're looking at are PCBs and mercury, because we know they're the ones of greatest concern, and they're the ones that tend to drive the fish consumption advisories. And over on south of Cornwall Island, where you have you know, I've talked a lot about mercury, but when you get on the south side of the river, you start thinking about PCBs, and so that's really one of the things that they're looking at too. But Abe, it's not stopping there. He wants to look at a lot of different contaminants. So one of the things we'll be doing is archiving some of the samples, so that when the resources come together and the questions get asked, that there'll be further analyses that will be done. And Matthew, maybe you can fill in some of the gaps there for me.
You did a great job. Just to say that we've been sampling fish since September, and have a great team in place for that. The weather is getting colder out there, but we're out there most weeks, and we're using gill nets. So if anyone in the community is out and you happen to see gill nets set out in the river, that's most likely part of this project. And we've been setting them around Cornwall Island and St. Regis, and we'll be setting some further downstream to get a range of geographic areas where we might expect a range of different, possibly a range of different contaminant signals in fish if they are present. So that's, Jeff had mentioned some of the species of interest. They're all fish that people would be expected to consume, such as walleye and perch, smallmouth bass. We caught a lot of those, and huge lungers actually this fall. And northern pike would be another species that we're catching as well. So we take fillets off all these species and freeze those, and those will be sent to a government lab where they'll run the contaminant analysis on those fillets.
Thanks so much, Matt. I've got questions lined up for you that have come in, and they're really generated because of all the field work you do. And of course, for those people who follow Matt Wendell, you'll see that he's on social a lot, and there's questions that come through on social that people have asked. And so I have a couple of them here. Can I go ahead and ask you?
Sure, go ahead.
So, is there any way to prevent people from fishing Chinook salmon out of the mouth of Rotary Creek? And for those of you who don't know, the Chinook salmon are currently spawning in Rotary Creek, and it's a wonderful thing to go and see. So, what's your answer to that, Matt?
Rotary Creek's this great man-made feature in Lamoureux Park and Cornwall that actually one of our research scientists, Brian Hickey, was instrumental in helping create that. And it's a fast-flowing cold stream, and Chinook salmon are attracted to these types of streams in the fall. So they've been using this creek shortly after it was actually constructed to spawn in. They return every fall, and they're very vulnerable. The creek's very shallow, so these large fish, they're 5 to 10 pounds, they're very vulnerable when they come in to spawn. And actually, there's a no fishing regulation in the creek to prevent damage to these populations, or not just salmon, but other species as well. So if you do see someone fishing in the creek, please inform them that they're not supposed to be there. There is signage there. People might be fishing at the mouth of the creek where I guess they would be permitted to. So the best thing is just education with people. Just explain, if you see someone say, hey, look, these are fish that are coming here to spawn. This is part of their critical part of their life cycle, where if you catch them and don't allow them to do this, you won't have fish, in 3 to 5 years returning here and spawning again. So usually when people find that out, and this wonderful life cycle of salmon, they might be motivated to not fish them there. There's always going to be people that don't want to listen to that, but overall, there's an overwhelming sense of appreciation, I think, for especially Rotary Creek, such a beautiful location. So that'd be my suggestion, just to do a nice have a nice educational moment with somebody in a nice way.
Okay, so the next question we have is also to do with spawning, and it's about tagging fish. Do you ever tag fish to see their paths while spawning? So the question here was related to the sturgeon, but I guess it could be for any of the fish.
Yeah, we have done fish tagging in the past that has been focused on American eels and looking at their movements in the river. We do periodically do tagging of fish with little T-bar tags that are visible from the outside of the fish. And if people are angling and they catch one, there's some information on the tag that you can report back, and we get information on how much that fish has grown and where it's actually moved. So we do try to tag a lot of fish that we do catch and release at the Institute with these types of tags. So if you see one of those, please get in touch with us. And there's other organizations out in the river as well on the US and Canadian side that are also tagging fish as they're released. And there's a big program on stocking sturgeon in the St. Lawrence River and Lake St. Francis that's led on the US side and also by OPG. So you might see some tags on sturgeon as part of that program as well.
Thank you. We've got some more questions. I was wondering if there was anyone else who wanted to add anything on the questions I've just put to Matt. Everyone's doing okay? There's a question about when does the salt water from the ocean join the fresh water of the St. Lawrence?
That's a very good question because it is actually something that has been changing over the last couple of decades. Usually we used to think that the saltwater signal was the location was close to Quebec City. Sometimes people were saying the bridge, the main bridge of Quebec City. But now it seems that we do see times of the year where actually the salinity is going up upstream of Quebec. And that is certainly something to watch in the future as the water level will rise in the oceans. And there will be a different dynamic in the estuary of the St. Lawrence that is going to bring more salt water going upstream. In the long term, and as scientists, we don't want to be too alarmist, but the reality is that if we look at the scenarios for water levels in the oceans, things will get a lot saltier inland. And it is a problem going upstream of Montreal because many municipalities are using the water from the St. Lawrence for agricultural purposes and also for drinking water purposes. So all of these municipalities and activities will be impaired in the future. So that's not a too good story here. And I will add others to add.
Thank you for answering. When I read that question, I was thinking it was going to be a simple, straightforward answer, and then Jerome jumps on and makes sure that we understand that all these things are complex. We've got sort of 2 minutes left before I need to wrap this up for the end of the session. There's one question here which I guess the person who asked it would probably like it to be answered, so I'll put it out there. How does mercury biomagnify in fish? Someone willing to take that one on?
Sure, Allison, are you interested in going at that one?
I think I'm going to leave it with you. You're more of a mercury expert.
Okay, well, we'll get started, and I'm sure there's a number of people who could chime in, and so that's a really great question. And so what happens is mercury, the form of mercury that biomagnifies, it's not just straight plain old metallic or even ionic mercury that we think about. It's mercury that gets transformed by bacteria, often which live in sediments, to methyl mercury, and they think it's bacteria are doing that as a way to detoxify the mercury that they're coming in contact with. The ionic, we call it a 2+ ionic form of mercury that gets methylated to methyl mercury. And it's that methyl mercury that, as it gets produced by the bacteria and then makes its way out in either into benthic invertebrates that are living in the sediments. So these bacteria are really fascinating because they need to have certain conditions in order to be present. These methylating, they're actually these methylating bacteria, they're sulfur reducing bacteria that often lives just below the surface of the sediments. And then that methyl mercury finds its way and starts to bioaccumulate into benthic invertebrates. And it turns out that methylmercury, when it's ingested, binds very well to proteins. And so that's why it ends up in staying in the tissue of these benthic invertebrates. And of course, then at that point in time, once it's into benthic invertebrates, which is the base of the sediment food chain, and it gets sediment foraging fish, will eat the benthic invertebrates, and then you have this chain reaction of the food chain that happens, and through this biomagnification process. And what it means is that every time that you go from one species to the other, you actually get a multiplier effect. So it may be very low at one stage at the very base of the food chain, but as you reach the top predator, something like a pike or a heron or an osprey, you get into very high concentrations, and in the highest levels, we know methylmercury is a neurotoxin, and then starts to have these harmful effects on species.
Yeah. Thank you, Jeff. I'm cutting you off a little bit because I've noticed that I missed a question that came in from Nick Cox, and I want to squeeze it in before the 12 o'clock comes around. So Nick is asking, there were several references to the ongoing decline in fish concentrations in lakes in Francis, particularly the reduction in size, a number of yellow perch. Is this a result of zebra mussels, effort to reduce phosphorus, or something else? My question is, what is the impact of the loss of pulp mill effluent? Pulping discharges half the tree to the river in the form of carbohydrates. Since 1970, there was a gradual reduction of over 200 TPD BOD equivalent. So he's giving some statistics there, and it's something I know Nick is curious about, and he brings up now and then. So is there someone who would like to take that on? Anyone? We've only got a minute, so it'd be quick.
I think, Jeff, you have to start us on this one.
Well, I guess I'll start it. And it's interesting that it talks about carbohydrates, and obviously we think of those as bacterially active. So the process of respiration and that in the river and turnover and just turnover of activity. So, I mean, he's asked this question before. I'll unfortunately I'm going to disappoint Nick again, because I don't necessarily have a great answer to that question in terms of saying I know exactly what's going to happen. But I think the more likely is the effects are associated with the nutrients that were associated with those discharges, and certainly they were important, significant, because the mill was a significant discharger into the area, and those nutrients are part of that whole question we talked about earlier about the reduction of nutrients since the 70s. So I think that's probably the most important aspect of it.
Okay, thanks, Jeff. And sorry to rush that one. I looked into my inbox and saw that I'd missed that question that had come in amongst about 50 new registrations for the symposium. So apologies, Nick, that came at the final hour. I want to take a minute just before we sign off to thank our panelists. Thanks everyone for joining us. That was really great. Of course, I've never done this before, and so I wasn't sure how it was going to go, and I found all the answers really interesting, and it was great to have you here. So thanks for coming in. I want to say to everyone who's watching, please jump off this live event and join us again at 12:15. Jerome, Marty, and Patrick Nadeau are going to entertain us over lunchtime, and we will also be making our River Award announcement. So please come back on, go and grab some lunch, and join us again in 15 minutes. Okay, bye-bye.
Bye, thank you.